Science speaks the language of numbers, but behind every number is a life. We sought the keys to longevity in labs, but the answers were right beside us. Our dogs, biological sentinels sharing our rhythm on a compressed timeline. Their DNA proves their aging is a projection of our own lives. Welcome to Mega Say!
Welcome, expert colleagues. As professionals in medicine, neurophysiology, and biogerontology, we all know the fundamental bottleneck in human longevity research, right? Conducting lifetime clinical trials in human cohorts is, frankly, practically impossible. And our traditional laboratory models, short-lived invertebrates and highly inbred rodents, well, they simply lack the genomic diversity, the environmental complexity, and the spontaneous multimorbidities that actually define human senescence. So today we're examining the rigorous data that positions the companion dog, Canis lupus familiaris, as a vastly superior translational model.
Here is our roadmap for today. We're going to move systematically, starting from macro-level demography, zooming all the way down into molecular pathology, and finally wrapping up with clinical pharmacology.
So, let's kick things off with Section 1, the mathematical alignment of actuarial aging dynamics.
Okay. To really validate the companion dog as a predictive model, we have to look at the Gompertz hazard function. You're intimately familiar with this. It defines the exponential increase in mortality risk as an organism ages. But look closely at the Gompertz slope comparison here. You'll notice the rate of demographic aging, that slope B, is actually mathematically lower in companion dogs. So why the compressed 10 to 15-year lifespan? Well, it's driven by a substantially higher initial baseline hazard, or A. This perfectly reflects the intense biological costs of their rapid early development and those extreme breed-specific physical variations.
And that high initial baseline hazard perfectly sets the stage to observe something really fascinating: the actuarial impact of clinical intervention. When we pull data from massive providers like Trupanion and Agria, we see that dogs enrolled in health insurance before 2 years of age actually live 1.5 to 2.0 years longer. Neutered, insured dogs are hitting a median lifespan of 12.2 years, compared to just 10.5 for uninsured groups. It's a massive shift. It proves that regular, proactive diagnostic access fundamentally alters their biological aging trajectory, literally mirroring human preventive medicine and validating the One Health concept for our models.
Moving right along to Section 2, Biobanking and Metabolomics. Let's see how this all translates to biological age on a molecular level. Thanks to massive initiatives like the Dog Aging Project Precision Cohort and the Mars Pet Care Biobank, we finally have the deep molecular profiling we need. What we're seeing is a progressive, systemic decay in canine plasma metabolomes that highly correlates with human mortality markers. Just look at the accumulation of long-chain acylcarnitines. This signals a critical failure in mitochondrial beta oxidation, basically a total loss of metabolic flexibility. Concurrently, we see a spike in post-translationally modified amino acids, which points to compromised lysosomal clearance. It's a severe failure in cellular proteostasis.
But here is the real kicker: a stark decrease in bioenergetic precursors. The systemic depletion of nicotinamide absolutely cripples the NAD plus salvage pathway. Without that primary substrate, NAD-dependent sirtuins simply cannot regulate DNA repair or mitochondrial biogenesis. It's basically human metabolic collapse playing out in real time, driving the onset of physical frailty in both dogs and humans.
For our neurophysiology experts tuning in, let's jump into Section 3 and examine canine cognitive dysfunction syndrome.
Aging companion dogs spontaneously develop CCDS, and it shares major histopathological hallmarks with human Alzheimer's, specifically progressive beta amyloid accumulation and glymphatic clearance disruption. But notice this absolutely critical structural distinction. Advanced human AD features highly structured, dense core fibrillaire plaques, right? But canine amyloid deposits remain primarily diffuse, unstructured oligomers. This is actually a huge advantage. It makes the canine brain an incredibly accurate model specifically for the early amyloid-driven phases of the disease, letting us test translational neurotherapeutics way before irreversible parenchymal destruction sets in. And we can diagnose this ante-mortem using the DISHAA behavioral framework. This isn't just subjective behavioral observation. These clinical signs map directly to specific regional atrophy.
Sleep disturbances point right to the degeneration of the suprachiasmatic nucleus. Activity changes reflect striatal dopamine depletion. For us researchers, this is a game changer, because these signs are entirely verifiable via advanced 3D MPROG MRI sequences, allowing us to quantify diffuse cortical atrophy with pinpoint precision.
All right, shifting gears to Section 4, Systems Immunology and Comparative Oncology. The evolutionary conservation we see here is just striking.
Digging into the specific cellular drivers, data from controlled environments like the Vika sled dog project reveals a severe myeloid lineage skewing in the bone marrow as these dogs age. This leads to a massive accumulation of PMMDSCs, or polymorph nuclear myeloid-derived suppressor cells. This is the classic inflammaging cascade. These cells are highly immunosuppressive. They inhibit T-cell activation, basically blinding the adaptive immune system to novel antigens. And naturally, this promotes a highly pro-tumorigenic microenvironment.
And this highlights the incredible power of comparative oncology. Because companion dogs share our homes and have fully functional immune systems, they spontaneously develop malignancies with molecular architectures identical to human patients. Canine osteosarcoma shares the exact same TP53 and RB1 genetic alterations as pediatric osteosarcoma. Mast cell tumors are driven by KIT mutations, just like human gastrointestinal stromal tumors. It allows us to co-develop targeted therapies, like tyrosine kinase inhibitors, in a real-world, complex immune context. You just don't get that in a petri dish.
Let's move into Section 5 and explore how this pathophysiological alignment actually facilitates the clinical translation of novel geroprotectors.
The sheer scale of veterinary trials happening right now is unprecedented. We're evaluating small molecule modulators under rigorous double-blind parameters. Take the Triad trial. They're using low-dose rapamycin to target MTRC1 inhibition in 580 dogs. Then you have Loyal's Therapeutics. Their STAY study is testing LOI-002 on a whopping 1,300 senior dogs to preserve insulin sensitivity. Meanwhile, LOI-001 and 003 are targeting the IGF-1 axis, which we know drives accelerated biological aging in large breeds. This is a monumental shift from reactive treatment to true proactive longevity medicine.
And this momentum is pushing straight into gene therapy. Myxomatous mitral valve disease, or MMVD, inevitably leads to congestive heart failure. But Rejuvenate Bio's RJB01 hits it with a dual-vector AAV delivery system. First, it upregulates FGF21 to boost systemic metabolism. At the same time, it drops in a decoy receptor, STGF-beta-R2, which neutralizes circulating TGF-beta1. This completely halts cardiac fibroblast activation. In clinical trials with Cavalier King Charles Spaniels, this dual approach delayed advanced heart failure progression by over a year and a half compared to standard care. That is a massive breakthrough.
Finally, in Section 6, let's weigh the methodological framework and, frankly, the economic realities of these models.
When we design longevity trials, we're constantly fighting trade-offs. Highly standardized laboratory rodents, like UMHE3 mice, have their place, but companion dogs provide massive genomic diversity and true environmental exposure, stuff you just can't replicate in a pathogen-free vivarium. Furthermore, and this is critical, dogs offer a path to the FDA's Expanded Conditional Approval, or XCA pathway. This allows drugs to achieve conditional marketing authorization based on a reasonable expectation of effectiveness.
And to put that into perspective, the financial disparity is just stark. Because rodent mortality increases exponentially, the cost to generate surviving healthy late-life mice goes through the roof. The estimated cumulative cost to maintain a single tumor-free 32-month-old laboratory mouse is roughly $1,400. One mouse. With companion canines, the baseline procurement and daily husbandry costs are entirely absorbed by highly engaged pet owners. This community science paradigm means your entire trial budget goes exactly where it should: advanced diagnostics and molecular screening.
So I'll leave you with this final provocation. As experts shaping the future of geroscience, consider how leveraging this established veterinary regulatory pathway, combined with the profound biological alignment of the companion dog model, could completely transform the temporal and financial scale of your next human longevity trial. The tools are here. The pathway is clear, and by deploying these models, we can absolutely accelerate the clinical translation of next-generation therapeutics.
Thank you for being with us. MegaSci is for you and for us.